Hey everyone, welcome back. In today's video, we're going to talk about FASTQ files, one of the most common file formats in genomics and bioinformatics. So what is a FASTQ file? It's a file format for storing raw sequencing data. Raw meaning the data was just generated and hasn't been processed yet. Now FASTQ files typically contain information about DNA or RNA sequences, and you'll usually see them with a .fastq or .fq file extension. So where do FASTQ files come from? So they're generated by sequencing instruments, large, high-tech machines like the Promethean, NovoSeq, and Revio. These instruments are built by companies like Oxford Nanopore, Illumina, and PacBio, and can cost hundreds of thousands of dollars to purchase. And the basic idea is that you feed them genetic material, and they output FASTQ files, a digital format we can analyze. And each entry within a FASTQ file represents a single read, a short sequence generated by the sequencer. Remember this term because it comes up a lot. And each entry within a FASTQ file is made up of 4 lines. The first, a unique identifier, starting with an @ symbol. The second, the nucleotide sequence that was generated by the sequencing instrument. Third, a +sign character. And fourth, a line of Phred quality scores, which indicate how confident the sequencer was in identifying each base from the corresponding nucleotide sequence. So now that we understand the format, let's look at a real FASTQ file. Now it might look a little intimidating at first, but take a moment, pause the video, and try counting how many reads you see here. If you counted 4, you're absolutely right. Each read is 4 lines long, starting with a line that begins with the @ character. Nice work. You're already thinking like a bioinformatician. So what are FASTQ files used for? So they're the starting point for nearly all bioinformatic workflows, including quality control, sequence alignment, genome assembly, and many other downstream analyses. And if you don't have your own data yet, don't worry, there are plenty of places to download them. So where can we get FASTQ files? We can download them for free from online databases like the Sequence Read Archive. People usually refer to this as the SRA, the Gene Expression Omnibus, GEO, and the European Nucleotide Archive, ENA, among many others. And the cool thing about these databases is that they contain real data from previously published experiments. So you can try things like reproducing the results, testing new hypotheses, or even conduct a meta-analysis. And you might be surprised to learn how many organizations generate FASTQ data. Hospitals like the Mayo Clinic, government agencies like NASA, and public health institutions like the CDC all routinely produce sequencing data. In fact, the CDC and other US government agencies have been generating a large amount of sequencing data in the form of FASTQ files to study the recent outbreak of H5N1 influenza virus in dairy cows. And since influenza viruses don't typically infect cows, the appearance of a highly pathogenic strain on US dairy farms raised some serious public health concerns. And sequencing the viral genomes is helping scientists better understand how this happened and how it can be prevented from happening again in the future. Now that's it for this video on FASTQ files. In the next video, we'll be talking about one of my favorite genomic file formats, Pod5. I'll see you there.